and we're picking up on chapter 27 of Bitten, The Secret History of Lyme Disease and Biological Weapons by Kris Newby. DNA Detectives, Milwaukee, Wisconsin, 2018. Genetics is about how information is stored and transmitted between generations. John Maynard Smith, British theoretical and mathematical evolutionary biologist and geneticist. I rode the glass elevator down inside the cavernous atrium of the Hyatt Regency Milwaukee in June 2018, below the 280 or so attendees at the annual meeting of the American Society for Ricketsology milled about the breakfast buffet, excited to share their insights into the wily, fascinating rickettsial organism at the center of this mystery. The first presentation, delivered by Ulrich Uli Munderloh, an entomology professor from the University of Minnesota, with a doctorate magna cum laude in veterinary medicine from Ludwig Maximilian University of Munich. She jogged onto the stage wearing a rumpled plaid flannel shirt, cropped cargo pants, and dusty running shoes. She had gray blonde hair and was wearing a sterling manta ray pendant around her neck. Munderloh began flipping through slides of rickettsia genetics at a blistering pace. Her research focus was on anaplasmosis, a disease caused by a rickettsial organism that invades white blood cells. The organism has 1,210 genes, and Munderloh seemed to be on a first-name basis with most of them. She shared tips with the audience on ways to hack into the organism's genome, to insert fluorescent markers so biological processes could be watched under a microscope, and she discussed strategies for knocking out specific gene sequences so that researchers could figure out how these organisms cause disease, information that could help prevent infections. The audience sat in rapt attention. No one was checking their email. The meeting attendees were an international mix of microbiologists, veterinarians, military scientists, and public health officials. Their language was laced with acronyms like OMP, outer membrane protein, and MFP, membrane fusion protein. Their slides were loaded with mandala-like circular family trees of rickettsial species, pictures of folding proteins resembling bags of colorful gift ribbons, and schematics of rickettsia cell hacking strategies that looked like football playbooks. By the end of the first day, I learned that each rickettsia species has unique and specialized ways of breaking into and replicating in its mammalian host. The study of rickettsia is a field that requires patience and a large team, including a lab manager, skilled technicians, genetic bioinformaticians, senior researchers, and eager doctoral students willing to work long hours for meager wages. It might take weeks to grow rickettsia in sufficient numbers to start an experiment, and if just one contaminating organism gets into a growth medium of living cells, the process has to be restarted. This was a far cry from Willie's generation, when discoveries could be made only by one scientist staring into a microscope. Still, the new gene-altering technologies were enabling a much deeper understanding of differences in species and mechanisms of infection. From the beginning, I was amazed at how open and friendly the rickettsiologists were compared to the Lyme researchers. Unlike the Lyme sessions in the IDSA-sponsored conferences, the American Society for Rickettsiology welcomed journalists and science writers with open arms. The Rickettsia Society didn't have a public relations minder sitting by me when talking to researchers. There were no patient protests outside the hotel. The organizers hadn't posted extra security guards outside to keep patients away from the proceedings. And when I approached the CDC employees with questions, they didn't turn their backs on me or tell me to contact the public information officers at the Atlanta headquarters. I pondered why Lyme disease researchers were so much more paranoid than their Rickettsia counterparts. Thinking back on my research for the Lyme documentary, Under Our Skin, I concluded that there was much more money at stake with Lyme disease. It was the first major disease discovered after the Bayh-Dole Act at the Diamond versus Chakrabarty Chakrabarty Supreme Court decision, making it possible for the NIH, the CDC, and universities to patent and profit from ownership of live organisms. When the causative organism behind Lyme disease was announced, something akin to the Oklahoma land rush of 1889 began, as scientists within these institutions began furiously filing patents on the surface proteins of and DNA of the Lyme spirochete, hoping to profit from future vaccines and diagnostic tests that used these markers. For example, an NIH employee who patents a bacterial surface protein used in a commercial test kit or a vaccine could receive up to $150,000 in royalty payments a year, and among that might double his or her annual salary. All of a sudden, the institutions that were supposed to be protecting the public health became business partners with Big Pharma. The university researchers who had previously shared information on dangerous emerging diseases were now delaying publishing their findings so they could become entrepreneurs and profit from patents through their university technology transfer groups. We essentially lost our system of scientific checks and balances, and this in turn had undermined patient trust in the institutions that are supposed to do no harm. With Lyme disease, there's no profit incentive for proactively treating someone within a few weeks of inexpensive off-patent antibiotics. Curing people is a sustainable business model. It's the patentable vaccine and mandatory tests before treatment that bring in the steady revenue year after year. I had two objectives in attending the Rickettsia conference: to learn as much as possible about the spotted fever rickettsia and to see if anyone had heard about Willie's mysterious Swiss agent USA. At every coffee break or meal, I'd work the room reading name badges and asking questions. I tried to hang out with the Rocky Mountain Lab researchers as much as possible. By the end of the conference, I couldn't find anyone who had heard of Willie's Swiss agent USA. I spoke at length with Lee Fuller and found the Fuller Laboratories, which specializes in developing tick-borne disease diagnostics. He explained how hard it is to test for rickettsia in blood and said, if you're not looking for it, you won't see it. I cornered David H. Walker, MD, a pathologist and the director of the Centers for Biodefense and Emerging Infectious Diseases at the University of Texas Medical Branch in Galveston, and asked him what he knew about Rickettsia helvetica. He said he hadn't heard of its being found in the United States, but he thought that the European-based organism was very rarely pathogenic to man. On the last morning, I had coffee with Michael L. Levin, a researcher in the Rickettsia zoonosis branch of the CDC. I didn't know it at the time, but he was about to publish an article that would challenge a famous Rickettsia axiom, a supposed fact accepted by Willie and other researchers. That axiom stated that when a mild and virulent rickettsia mixed inside a tick, the mild species prevented the pathogenic rickettsia from being passed on through tick eggs. I highly respect Willie Bergdorfer as a brilliant scientist, for his pioneering works, said Levin, but some of Willie's axioms are not reproducible. Levin attributed this error to the fact that newer technologies like PCR were not available to Willie and his colleagues. Polymerase chain reaction, or PCR, is a method for producing many copies of DNA segment and making it easier and faster to detect low levels of bacteria. Then he chided the scientific community for not verifying the long-held belief on rickettsia infections, writing, "Yet this hypothesis was accepted by the scientific community as a proven fact, and after numerous reiterations in scientific papers, reviews, and textbooks, it became one of the best-known axioms in rickettsiology." Levin's revelation was important to the Swiss Agent USA discovery, given that Willie had often referred to Swiss Agent USA as a harmless symbiote that would reduce the transmission of virulent spotted fevers to the next generation of ticks. Levin, however, had found that multiple species of rickettsia can be transmitted through a single tick, a situation that could complicate diagnosis of human disease. I left the conference with the feeling that rickettsias can be deadlier than more chronic than Lyme disease, yet once Lyme disease was discovered, this threat had been ignored. 2 months later, after immersing myself in the medical literature on rickettsias, I found an unnamed forgotten rickettsia strain that I thought might be Swiss Agent USA, labeled with Willie's initials, WB82. It was buried in a 1998 journal article describing a strange bastard of rickettsia found in Missouri. It looked like Rickettsia montana, now called Rickettsia montanensis, but didn't seem to be related to any of the American strains. I called one of the authors of the 1998 article, Susan Weller, PhD, director of the University of Nebraska State Museum. At the time, she was proofreading the placards that would be used in what she called the second largest collection of parasites in the Western Hemisphere. It featured a life-size model of a 100-foot whale tapeworm. Weller's specialty is the evolutionary biology of insects, and in 2017 she was elected president of the Entomological Society of America. So how did a nice girl like you ever get interested in a parasite like Rickettsia WB8-2? I asked her. Weller told me that a few years ago, Uli Munderloh, a keynote speaker at the Rickettsia Society meeting, had come into her office and asked if she would help solve a genetic mystery. Munderloh and her colleague Timothy Curti had grown and examined a rickettsial species discovered by Willie, WB8-2, and had isolated another of these strange rickettsias, MOAA. But they couldn't figure out where they'd come from. They asked Weller to analyze their genomes in the same way that 23andMe decodes the ancestry of human beings, by sequencing parts of the genome and tracking back the genes that had been passed down from generation to generation. Weller accepted the challenge and ran a genetic analysis, and she discovered a puzzling thing. While the bulk of the rickettsia genomes were closely related to R. montana, now R. montanensis, parts were from an old world species, not a North American one. Under natural evolutionary processes, this type of old world / new world genetic mashup would be a near impossibility. Because these strains didn't seem to be overly harmful to humans, they were ignored for 20 years. Finally, in 2016, the CDC sequenced WB8-2's entire genome and declared that this Missouri rickettsia's nearest relative was a native Far East Siberia named after the city of Kharbarovsk. MOAA was so closely related to WB 8-2 that they were both named Rickettsia emblyomi, later renamed Rickettsia emblyomatis. Both are carried by Lone Star ticks. When I summarized the clues that Willie had left me on Swiss Agent USA, Munderloh guessed that it was probably Rickettsia buchneri, a close relative of spotted fever rickettsia that were not infectious to humans, perfectly adapted to living inside deer ticks. Her colleague Curti had found that Rickettsia buchneri was a symbiont that happily coexisted inside ticks because it produced the B vitamin complex needed for the tick to extract nutrients from mammalian blood. This left me with evidence that there were many new foreign DNA rickettsia being discovered across the United States. I also had expert opinions that Swiss Agent USA was a harmless symbiont, that the disease-causing tick rickettsia could coexist with harmless ones in a single tick. To determine whether Swiss Agent USA was contributing to the Lyme disease operation, to determine whether Swiss Agent USA was contributing to the Lyme disease problem, the pathogen would have to be identified in blood samples of sick Lyme patients from the 1970s and today. Finding a freezer full of unusable blood samples from 45 years ago would be nearly impossible, but I had an idea to get present-day samples tested. Chapter 28. Stanford University, California, 2014. Never doubt that a small group of thoughtful, committed citizens can change the world. Indeed, it is the only thing that ever has. Attributed to cultural anthropologist Margaret Mead. A receptionist ushered me into a physician's office, in the Department of Infectious Diseases and Geographic Medicine at Stanford's School of Medicine. I was there to discuss the first draft of an article I'd written about the physician in Stanford Medicine magazine. In the piece, I tell the story of one of his patients with a mysterious condition that caused symptoms similar to Lyme disease. He and his team were the first to identify immune system molecules in the blood that are unique to the condition. The first hard evidence that these people had He and his team were the first to identify immune system molecules in the blood that are unique to this condition. The first hard evidence that these people had a real disease. With this information, a diagnostic test could be developed, enabling objective evaluations of potential treatments. I had a great deal of empathy for these patients, but I was happy to write the article. Their symptoms are largely invisible, and with no gold standard tests or treatments, physicians often have to turn these patients away. So I poured my heart and soul into the article and created a video to go with it. I also knew this physician as Dr. D, the senior attending physician who in 2003 told me and my husband that he didn't think we had Lyme disease. As I sat in his office, I wondered if he remembered me, but I didn't mention our previous meeting. No sense in dredging up that traumatic encounter, and all was forgiven on my end. More than a decade after the tick bite that changed my life, I had a deeper understanding of the Lyme problem from a scientific, political, and policy point of view. I knew that the infectious disease departments at most major medical centers, including Stanford, were simply following the iron-fisted IDSA Lyme guidelines that state that chronic Lyme isn't an infectious disease and that it can't be treated with long-term antibiotics. If Dr. D had kept up on his patients, if Dr. D had kept us on as patients, He might have been reprimanded or even fired, and to his credit, he was the first to test us for Lyme, and that ultimately put us on the path to wellness. As I sat in his office that day, I began thinking of him as a potential ally. We both cared a great deal about patients who had fallen through the cracks of the medical system, and I knew he'd spent years collecting blood samples from about 600 patients from the United States, some potentially with tick-borne diseases. A subset of samples was sitting in a freezer somewhere at Columbia University, awaiting a genetic screen for viruses or bacteria that might cause their chronic symptoms. At the end of the meeting, I took a chance and asked him, "Are you screening for any rickettsia?" He said he didn't know. The genetic sequencing was being done at Columbia University in collaboration with Ian Lipkin, PhD, a famous virus hunter, and with tick-borne disease expert Rafael Tokars, PhD. Dr. D opened the study protocol on his laptop and realized that there were no rickettsia on the screening list. I quickly laid out the story of Willie and the Swiss Agent USA, and Dr. D said he'd see if rickettsias could be added to the search. As I got up to leave, he added, "When you came to my clinic before, you weren't allowed to treat chronic Lyme disease. It was department policy." I'm sorry. At least he said he's sorry. A few months later, I was passing through the Stanford Hospital when I saw Dr. D walking away from me about 30 meters down the hall. I called out to him as he swiveled around. "Congratulations on your story on the immune system," I said. He had just published a new study that had identified inflammation molecules that could be used to identify patients with this condition. The news had been picked up by national media and was being featured on the medical school's homepage. He smiled, ran up to me, and enveloped me in a hug. "You were the inspiration for this research. Your courage in going against the establishment inspired me." Then he thumped his heart with a fist and said, "This is for the patients. I want to do the same as what you're doing for tick-borne diseases." As I was finishing up this book, I contacted the Columbia researchers to check on their progress. Tokarts, who was doing a rickettsial search within ticks collected around Long Island Sound, thought he'd be done with the analysis by the summer of 2019. Lipkin, who was screening Dr. D's blood samples for rickettsia, wasn't clear on his timeline. He said the key is in joining results of tick screens with serology. The presence of rickettsia in ticks does not necessarily mean they can be transmitted to humans, replicate, and cause disease. We are trying to rigorously pursue this line of research, difficult to do without financial support. While I was pleased that someone was finally looking for ricketsias around Long Island Sound, I wished there were more of a sense of urgency. After all, every day we delay, 1,000 more people are bitten. And we'll leave it there for today.